All right, great, thank you. All right, we're going to move on. So our next speaker is Dr. Chris Chabot. Dr. Chabot is a professor of neurobiology, physiology, and behavior at Plymouth State University in New Hampshire, and has been investigating horseshoe crab physiology and behavior for nearly 30 years. He is particularly interested in the mechanisms and adaptive advantages of horseshoe crabs' biological clocks and the interactions of these clocks with environmental cycles such as solar, tidal, and lunar cycles. His various grants and publications have focused on horseshoe crab biology from the genetics to the population level. The spawning survey data that he will report on involved a long-term collaboration with Wynne Watson, who recently retired from UNH, and many other scientists, organizations, and dozens of volunteers spanning more than a decade. So Chris, go ahead and take it away.
Very good, thank you very much. Can you see my main screen?
We see your speaker view.
Very good, thank you. I can't tell. Well, thanks very much, Derek and all, for inviting me to give a talk. Really appreciate it. And thanks for a great talk as well, Dave. I'm going to pick up your points a little bit later on. So we've been doing surveys in our population for over 15 years or so, and happy to report on some of those findings today. This has been a collaboration, big collaboration between Wynne Watson and I, of course, has already been mentioned, but also with Great Bay National Estuarine Research Reserve, Chris Peter, Lauren White, and Melissa Brogel. So I'll be talking about several things today. I want to talk, first of all, orient you all on our study site in Great Bay, New Hampshire. And then I want to talk about, obviously, Great Bay horseshoe crabs, adults, and talk a little bit about the range that we see that they occupy up here, talk a little bit about their behavior and activity rhythms. And some of the main points that I want to make are over here in red, that there are population differences that we see. Dave, in one of his seminal papers earlier on, showed that there are genetic population differences, and there are also behavioral differences. Next, I want to talk about spawning habitat. Of course, beaches are involved, but marshes are key, as Dave mentioned earlier as well. And then some of the factors that are involved in spawning, which we all know about. A couple of major points is up here, there are no significant day-night differences in general, and moon phase does not play a role either. And as well, the many volunteers that we have, it is so important, as Dave mentioned, to have a lot of volunteers to do this. And we do have a lot, and we can essentially monitor 6 different sites up here every day of the year, including a week before, or every day of the spawning season, including a week before spawning season starts. So we know when it does start. And lastly, I want to talk just a little bit about horseshoe crab development. After all, that does occupy 75 to 90% of their life. Talk a little bit about eggs, larvae, and some of the work that we've done in juveniles. And again, we see some population differences. And then summarize the work. Talk a little bit about the future work. So our study site is up here north of the Cape in this area in southeastern New Hampshire. This is a blow up of the region. This is our site made up of the Great Bay Estuary, which includes Great Bay and Little Bay. And this is the Piscataqua River. And it's about 9 miles or so out to the ocean. This is New Hampshire's largest estuary, about 6,000 acres or so. And it's made up of mud flats, salt marshes, eelgrass beds, and very limited beaches. There are probably around less than 1%. And these beaches, we'll show pictures in a little bit, are not the typical sandy beaches in general. They're made up of fairly large coral. Importantly, something I want to mention is there's very minimal fishing pressures up here. There's essentially virtually no harvest of animals. So this is from essentially an unfished population. This next slide shows a blow up of, or what I want to talk about now is the behavior of these animals, and just very briefly about their annual range. what we did to measure their range out in the estuary is we put these hangers on them and tag them, and then we could let them go, let them free roam, and then we track them. And this next slide is a blow up of one of the tracks from several weeks. This is from Megan Owing's work and ours back in 2019. And you can see that this animal was captured here and let go, and it spent quite a while on mud flats out here eating. This is mostly post spawning, by the way. And then by the end of the year, when it got colder, October, this animal went downstream, which is pretty typical. And by downstream, I mean out towards the ocean, but did not leave the bay. And when we look at a lot of these animals, Sue Schaller and Wynn and I did this work, mostly Wynn and Sue, obviously, did this work back in 2009. And what we see essentially is in the spring, these animals come upstream from their winter grounds. A lot of the animals hung out here near the Piscataqua River, came up in here and went through Great Bay. And then in the late summer and fall, they come back down, go deep, and essentially don't move from November to March. So right now, all of our horseshoe crabs up here are down deep in the mud and not doing much at all. So that's their annual range. And importantly, they don't go out of the estuary and enter into the bay, the ocean. And that's going to be different, obviously, than other populations. Another aspect I want to bring up here is that they have endogenous internal rhythms that are driven by clocks, endogenous clocks. And quite a few papers by Wynn and I in the lab and in the field. I did much of the lab work. Wynn did much of the field work, in fact, all of it early on with his students. And we have data that shows that they have these clocks. And how did we measure these behaviors? Well, very briefly, in many cases, we actually put them on running wheels, which sounds kind of bizarre, but it worked very well. Put individual animals in these running wheels. They would move these wheels, and every time the magnet went past this sensor, it would trip a switch and send data to our data collection system. And then we organize these data into what are known as actograms. And I want to spend a minute or so talking about these actograms so you're well oriented. This will show the behaviors of these animals over a month and by time of day. So you'll notice that this animal here was in a light dark cycle. And when we put the animal in up here, it didn't move for a bit, but then it started moving. So this is day one, and it was put in the running wheel here, and it starts to move here, as it turns out, during the day. Day two is plotted below it, and these data are double plotted simply so you can better see the rhythms and the organization of the patterns. So on the x-axis, we actually have 2 days worth going from 0 to 24 to 48. So this is day 1 to 2, day 2 to 3. So these data over here are exactly like you exactly double plotted data from over here. So you can see patterns that are very clear. And what you see is 4 bouts of activity, 1, 2, 3, 4 bouts of activity over the course of 2 days. And that means there are 2 bouts of activity over the course of 1 day. And these are, of course, our circotidal rhythms. The tides come in and out in our area of the eastern seaboard twice a day. And but this animal is in the lab in a light dark cycle, and but constant water conditions. So these rhythms are endogenous, and these are endogenous circotidal rhythms driven by an endogenous biological clock or clocks. These patterns really don't change much if they're in light dark cycles or in dark, the lack of a cycle, essentially. So that one of the points we would make from our population is that at least some or many of these animals I don't care whether it's night or day, they're going to move according to either their internal clock or we'll see in a few minutes the tides. So in the lab and in the field, we've actually deployed these running wheels in the field as well. And we've used a variety of other methods to look at their behaviors in the field. And this is what we see. In the field, of course, we see that they are coordinated with the tides. They are generally active during high tides, but some of them are active during the beginning of high tides, some during a later part of ebb, essentially, right? So that's our population. What I want to do is just talk very briefly about some of our work looking at other populations and the effects of environmental factors on these rhythms, these patterns of activity that these animals show. And of course, what I'm building towards is that these internal clocks are going to help them with their spawning tidal rhythms. So I want to show first some data from New Hampshire. And this is a lot smaller, but I just want you to see the patterns of activity. And you can see 1, 2, 3, 4 bouts of activity over the course of 2 days. So we are back to showing a different animal, but with 2 bouts of activity a day. These are circa tidal rhythms. And again, no light-dark preference. Over here is a periodogram analysis, a statistical analysis showing what the significant peak is. And in this case, it's 12.4 hours, again, a circa tidal rhythm. And what I'm going to show now is in the lab, we gave them different tidal frequencies to look to see whether or not these frequencies could affect their behavior. Because up and down the seaboard, from here down to the tip of Florida, in general, there are 2 tides per day, but on the Gulf Coast of Florida, which I'm going to show you data in a minute, there's actually only 1 tide per day. And in the Indian River Lagoon in Florida, there's actually no major tides per day. So in any case, when we exposed this population of animals from New Hampshire to 1 tidal bout per day, we raised and lowered the water once per day, essentially. We still see 2 bouts per day. They don't care that there's only 1 high tide per day. They are still moving twice per day. And that happens whether we give them, again, no tides or 2 tides. They don't care, at least our population up here. They show circa tidal rhythms. Well, what happens when we collect animals from Florida and put them in these running wheels as well? And these are animals from the Florida Gulf. And make a long story short, you see they have 1 bout of activity per day. Their periodicity is about 24, 24.8. It varies a little bit. And it doesn't matter whether there's no tides or whether we give them 2 tides per day, 1 tide per day, or again, no tides. And so what we have shown, and Becky Anderson in my lab did this nice work, is that Florida Gulf animals show 1 bout per day, and they actually have a diurnal preference. So if you look at the activity, you can see most of the activity takes place during the day, not during the night in these Florida Gulf crabs. And an interesting thing happens when the tides run into the dark is they do something we call switching. They switch from 1 bout, or what we think of as one of their internal clocks, to the other internal clock. That's more of a nuance. You can let that go. Happy to talk about it later or via email or what have you. But what this demonstrates pretty conclusively is that actually horseshoe crabs have 2 clocks, 2 circalunadian clocks, and that helps them to synchronize with the tides. So Gulf Coast animals have 1 bout per day, and they have a diurnal preference, as do Indian River Lagoon animals, but they are much more driven by the light-dark cycle. Not terribly surprisingly, because these animals are in an atidal or a microtidal environment. And so the take home here is that in horseshoe crabs, at least in the 3 populations that we've tested, they have endogenous clocks that drive these rhythms, and they're likely to drive their spawning activities during the spawning season, which up here is about a month and a half. But they're active here and likely elsewhere for much longer than that. And what others have shown, especially Lee et al. In 2010, Lee in 2010 showed, is that they're up on the tidal flats feeding, and their internal clocks are likely driving that rhythm. So with that slight discourse on other rhythms, or other populations, I'm going to go back to New Hampshire populations and look at their spawning, which is what many of us in the audience are primarily interested in here. Again, we see spawning rhythms in the field, and these data are from a couple of our papers. This is plotted in a slightly different way. These are counts looking at counts day and night over the course of in June over 3 days. And what you see is that we see actually this is distance travel data from animals that are tagged. And what you see is that they coordinate their activity generally out in the field with high tides, and not so much with time of day. Okay, so these are freely moving animals. And when these data are put together and analyzed, we really don't see a day-night difference. There is a tendency towards increased movement in the day in our population, but of course we do see a significant effect of high tide on activity. Not terribly surprising here. So tides are significant, of course. We've all seen that. We watch for their spawning during high tides, not during low tides. But time of day, again, in our population, not significant. What other factors are involved with field behavior? Nobody's going to be surprised by this either, but this is from Helen Cheng's really good work in 2015 in Wynn's lab. Waves, when there are no waves, we see more horseshoe crab on the beach than when there's 6 inches or less and 6 inches or more. We have a whole lot of data that we're currently analyzing. Wynn and Lauren White from Great Bay Near are heading up this effort. We should have statistics very soon. From Helen's work, rain has an impact. The day before rain, we see a higher density of animals on the beach. Day of rain, lower, and day after rain, a little bit less. We think that's probably because of salinity or the turbulence due to the weather event. So overall, waves and rain, there's a negative correlation of spawning, no surprise there. High tide, positive correlation, again, no surprises there, but maybe surprising for some of you who tend to do more of the nocturnal types of counts. We see no correlation, at least in our population, and likely, at least in the Florida populations, we would argue as well. What about habitat? Habitat matters. We horseshoe crab biologists have known for a long time that horseshoe crabs breed on beaches, and that is what most of the work in the literature has been published about. Really important work. Beach habitat is incredibly important. Up here, it's arguably whether we have beaches, but we call these beaches. A few southern folks would call these beaches. It's kind of tough on your feet, but nonetheless, we have these are kind of very cobbly beaches. There is good sand underneath and some mud, and you can see some horseshoe crab eggs in our beaches. But I also want to point out, as Dave made this really good point as well earlier, that recent work in Dan Sassen's, in collaboration with Dan Sassen and his and Michael Kendrick's shop down in South Carolina, marshes are also, as it turns out, important. Now, a lot of us have seen animals in marshes for a long time, but I think a lot of us, including me and others, have assumed that these are not great places for horseshoe crabs to lay eggs. So they probably aren't. They're just looking for other places. They're just what we call smurfing around, looking for beaches, and they kind of get lost. But we started to look at this, and Mike Kendrick and Dan Sasson looked at it first and published their paper in 2021. And Daniel reached out to me and Jennifer Matai, and now work with Margaret Conroy in Delaware, and looking at these marshes. And here's a marsh from Great Bay and a tidal creek. Here's some of my students out of the tidal creek. We're marking potential nests because we do see animals coming up into this area, and during low tide we see them hanging out in this area, and so we wanted to look to see whether or not they were actually laying eggs. So what are our methods? We beach counts, including on the marshes. We do egg sampling by using coring, and I'll show you that in a few minutes. And then later I'll show you a little bit about egg development and hatching, because Dave showed you very clearly data that suggests that oxygen concentration is really important, as is temperature. So take a look at that in a few minutes. So if you look at just the counts, and this is from Daniel Sasson's and a multiple author paper that came out very recently, 2024. We see a lot of animals in the marsh, and not significantly different than the number that we see in the beach. You can see a fair amount more of range here, though, clearly. So as it turns out, there's no difference in the two habitat types of the transects that we chose anyway. Both are used, no clear differences in density. Interestingly, there's a higher sex ratio across the three different sites in South Carolina and in Connecticut and in New Hampshire. Are eggs found in marshes? Well, they can be. Early on, when we were looking at this, we found a pair during low tide hanging out in a high marsh area. We lifted her up, and lo and behold, we saw eggs. How do we look to see whether or not they're actually using marshes? Well, we do corings every couple of meters, 50 per 100 meter transect. We do cores, and we collect these cores and look at the amount of eggs. And you can see here clearly there are eggs in the marshes, of course, and in the beaches as well. And the data from our 2024 paper are shown here. Again, lots of eggs in both. Again, in the marshes, a wider range, interesting. Okay. I'll come back and show a little bit of more recent data that we haven't published yet on this. But one of the major points here is that eggs are found in both. And if you look, you can kind of tell there's some development that's happening here as well, which is very interesting. A little teaser for a few slides later. So back to spawning accounts. What do we do up in Great Bay? Well, we have 6 different sites, and they are shown here around Great Bay. And we have a whole bunch of volunteers, and we also get out there and do some of these counts. And we count spawning pairs and singles. We do day counts only because in our population, we have shown pretty clearly that there might even be more animals during the day, but there certainly aren't more at night. So day counts are fine. And we do every day of the season. And I'll show you some of those data later on. We have done counts on breeding beaches for about 20 years, a few years early on, and then we've skipped a few, and then COVID and so forth. But we have a pretty good record, and I'll show you those data. For the last 3 years, we've been doing marshes, and we plan to continue to do those. And we have hundreds of volunteers over the time, and that couldn't have been done with Great Bay Near collaboration. So generally, we began this in 2012. We counted 50,000 crabs or more. Early surveys, we were able to, because of lower volunteer numbers, and we weren't collaborating with Great Bay at the time as much, we had 30 to 90% of the days of possible breeding season counted. But recent surveys and current 80 to 90% of our days at every one of our sites has some counts. And so recently 10,000 crabs per year with 70 volunteers or so, some of them shown at a educational session that we run every year, which we're due to run next month. So just a few metrics from our population. If you look at density measured in horseshoe crabs per 100 meters squared per day, you see a pretty good similarity across the several years. You will see a little bit of an uptick here, and that is likely because we added some of the marsh sites in the 3 years, and those happen to have some pretty high densities. But overall, in Great Bay, our population, according to our spawning count, seems pretty stable. Our sex ratios seem to be rising. There's no significant difference here, but we do see maybe a trend, and we're somewhat worried about that. This is curious because we do know, and we've seen from other populations, and Dave alluded to this earlier on, or outright stated it in his population, that the females early on were fished heavily. These are not fish. These animals are not fish, so we cannot tie this to fishing, this rise in sex ratio. So we're not sure what that cause might be. What do our data look like when we look at the timing? This is from 2023. And this is a pretty data-rich slide, so I'm going to take a few minutes here. This is in total horseshoe crabs, but that's okay. I'm going to show you the densities on the next slide. And what we see essentially is we have up here an early May start. These are actual numbers. We start on 1st of May, and these are all zeros. So people are out on the marshes and out on our transects, and we have zeros, and we see a nice early May start. And in general, here we see kind of a late to middle peak. Arguably, this is the peak, but it seems like for this year, for 2023, there might be 3 peaks. What are these different colors? These are our different sites, and Pickering Brook and Great Bay Farms are our marshy sites. And you can see maybe these are a little bit later. These are alternative habitats, we call them. And by late June, it's all over, certainly. We have a few crabs visiting, but most of the peak is within a month. And importantly, I want to point out here, there's no clear lunar effects. This is the new moon. This is the full moon. You could argue, well, maybe this is a new full moon peak, but I'm going to show you the last 3 years with these lunar cycles superimposed in the next slide. And I'm going to change the y-axis to the density, horseshoe crabs per 100 meters squared per day. And what you can see is last year there was one peak, and you might argue that it was close to the full moon. But 2023, the same data, 3 peaks, and 2022, that is not really close to the full moon. It's more in between full and new. So there's no clear lunar effects. We're not going to argue against them, but we don't see clear lunar effects in our population. And up here, folks, when we see the higher tides with new and full moons, what happens on our beaches is that the water rises so high that it ends up being up against either ledge or areas which is lousy for spawning. So keep that in mind. Our habitat is likely very different, especially compared to your very long beaches, where on your exceptionally high tides, some of yours anyway, you still have beach where they can spawn. at the high tide level. We don't. And what we see is as the water comes in, they tend to spawn, and I'm sure you've seen this as well, at the high tide line. That seems to give them a better purchase for digging, the females, because a little bit more gravity. All right, some more data. When are the first spawning dates over these years? It's pretty consistent. It's generally in mid-May, and that's what the x-axis is showing. Our first spawning dates defined as at least 2 pairs of horseshoe crabs on the beach. There is a weak, interestingly, correlation with the start time. There's a little slightly stronger with peak times and temperature R squared at 0.35s. Interestingly, for some of our sites, there's very strong R squareds for temperature, and it starts and peaks of around between 0.7 and 0.8, which means there are some seemingly outliers from some of our sites, and we need to do more data analysis there. Okay, I want to change this up a little bit and talk a little bit more about the development and tease a little bit some of our data, which will be in an upcoming publication. Here, what we're doing is we're looking at eggs in our beachy sites and in our marshy sites and staging them. Because some of you are thinking, sure, they lay eggs in the marshes, but boy, that's very anoxic. You can tell by the smell. It's, hydrogen sulfides. It can't be a great place for those eggs to develop. And I don't disagree. It seems like it wouldn't be. But what we see when we actually collect them and stage them, and these are data only from New Hampshire, but very similar data are seen in our other sites. If you look at the percent in these stages, early, middle, or late at a particular time point, this is about 3 weeks after being laid, you see a similar number in the early stages for beaches and marsh, and similar in the medium and late. So it does seem like animals are developing, the eggs are developing, and I won't say that they're hatching necessarily because we haven't done that work yet. That's certainly in the future. Want to talk a little bit about larvae and tiny juvies, first instar or second instar juveniles. In the field after they hatch, where do they go? There's some really nice data or studies done by Mark Botten and collaborators. These larvae, after they hatch, they are in the nest oftentimes for 2 to 3 weeks. And then they get up into the water column, especially around ebb, high tide. But they might be in the water column for 2 to 3 weeks. And other species, they get up into the water column, and that might carry them long distances to more favorable habitat. And that could happen in horseshoe crabs. We don't know for sure, but what Mark and his colleagues have shown is that there's a lot of these larvae and second instar juveniles in these mud flats adjacent to these areas. So it looks like they might use the tidal transport to transport them just a little bit of ways to the favorable areas for development for the next many years. Remarkably, some over winter. And we don't know if that happens up here. We're testing that. I had a wonderful email conversation with Dan Gibson. He was so kind to share data from one of his personal communications in the 92. This apparently happens in Cape Cod. We don't know if it happens up here. Some students and I are working on some data looking at vertical migration rhythms. Ellinger and Tankersley shows some beautiful data showing that groups of animals, larvae, do show vertical migration rhythms, and our system is a little bit different than theirs. We have individuals in these vertical water columns, 12 centimeters high, 2.5 centimeters wide, and we videotape them and look at their rhythms. And here's Jacob and Fletcher. This is a manuscript in prep. What we see very clearly in larvae are these beautiful vertical migration rhythms. I just wanted to point out actograms, and some of these animals show 1 bout per day. 24.2 hours for this animal. Others show 2 bouts per day, the more circadial rhythms. But remarkably, a lot of them, most of them show these circalunadian rhythms. And again, this is a manuscript in prep. As for tiny juvies, we do the same sort of thing with tiny juvies. We film them in these small 6-well plates. We have 96 of these at a time with 4 cameras. We can film, and Tyler Thomas and Wynn and I published a paper in 2020 showing some data from New Hampshire, Florida, and just want to point out that it's very similar when you look at the behavioral patterns of the second instars compared to the behavioral patterns of the adults from New Hampshire and Florida. That New Hampshire animals generally have circadial rhythms, whereas Florida generally have these daily or circadian rhythms. Now, what about juveniles 2 to 9 years? Helen Cheng did some beautiful work in Great Bay showing that they tend to be more nocturnal, like our tiny juvies do, whether they're in Florida. And I'm not showing the data from New Hampshire, but they tend to move during the night more than during the day. So juveniles are different, but I'm not aware of data from other populations, and that's a huge area of interest. Certainly, there's lots to do. This can have been done with a lot of volunteer involvement. Melissa Brogue has been great Benier as well. Lots of increased involvement since 2022. She has it working like a well-oiled machine, online signups and data submissions through a Google form. She provides wonderful weekly updates for our volunteers, and we have great turnouts. Here's Wynn at our orientation session, and annual wrap-up with volunteer feedback as well. And this year, we're really happy that Lauren White is helping out a lot with the data analysis, something we've really needed. A lot of next steps. Here are our sites that we use. 4 beaches in orange, 2 marshy in green, and the other multicolors are places we think there will be animals and eggs and breeding, but we have a lot of things to do. We need to describe these characteristics, and we're very interested in egg predation here, and habitat loss is key, obviously. So lots to do. Thank you all for your attention. Many thanks to all the volunteers, and I'll be happy to take questions.
All right, great. Thank you so much, Chris, for that. Couple minutes for questions. So we got a couple players. So let's do what we did last time. So start.
Yeah, talking about the Florida horseshoe crabs, can you just define diurnal preference?
Sure. When we measure activity in either tiny juvies, the second instar, or the adults, they tend to move more during the day than they do during the night, statistically speaking. If you look at those patterns that I showed earlier. And so they are essentially preferring, for the Gulf Coast animals, they prefer to move with the day diurnal high tide. For the Indian River Lagoon animals, they tend to move during the day because there's really no tides. Been down there. I spent a couple of weeks in Indian River Lagoon, and when you go out and try to collect the animals or the eggs, in the afternoon the wind picks up, and that's when they tend, and that the water tends to be driven up in some places. That's their high tide, if you will, wind driven. If it's too windy, they won't breed, as we've seen in other populations. But I hope that explains.
Yes.
I was wondering what kind of temperature and salinity ranges are the crabs living in and spawning in?
Yeah, great question. We've got lots and lots of data on both, and we're actively looking at those correlations. I'm gonna blow your mind with the salinity. So at times when you look at some salinity events in the winter or spring, it can get down to 2 or 4 PSUs. During that time, the animals are on the bottom of the bay, not moving very much. But with rain events in the summer, they can get down into the teens. It's not terribly unusual. Usually, the average is around 26 PSUs, and plus or minus what a few. In terms of temperature, of course, it gets down to 4 degrees C in the winter. And what we see in general in a different paper that I didn't have time to show you data on, we've shown that at 4 degrees C, animals don't move very much in the lab. They don't show any rhythms. They don't move very much. You warm it to around 11 degrees C, they start to move, but they really don't show rhythms. You get it up to 17 degrees C, and they do show those patterns of activity. In the field, we see with those correlations that I showed you, once the water gets up to 12 degrees, 11, 12, they're moving around in the field. They're probably not spawning until 15 to 17. The water gets a lot warmer, and it feels like bath water at times, even in Great Bay, during the peak of the breeding season when you're walking around. So that's up into the 20s.
Do we have any questions online?
Yeah, we have one question in the chat from Dr. Smith of if you were to allocate funds to habitat management for marsh and beach habitat, based on what you know now, how would you allocate those funds?
Oh boy, that's a great question. How much money are you giving me to allocate? So
a lot. We're giving you a lot of money.
Oh, thanks, Dave.
$1 million.
So yeah, that's not enough to unfortunately to so you're talking about allocating to research, I guess. A key thing.
No, this would be not research. This would be doing stuff, either buying the property, putting it into see 10 million.
Yeah.
Say you have 100 million. I don't care.
Right. So to your point, Dave, with global climate change and rising waters, I mean, we're going to lose a lot of the beaches. And so the refuge in many of our areas are going to possibly be the marshes unless the water, or when the water rises up even higher to those hardened areas. That's a tough question. Habitat protection is key in both areas. Clearly, the estuaries, the marshes, or the nurseries for many species, including, as it turns out, it looks like horseshoe crabs, but the beaches are key as well. So I'm not answering the question.
No, I heard what you were saying there. I agree. That's fascinating talk, and the work on marsh spawning is really really great. It's good stuff.
All right, great.